Good evening everyone. Today we start with the chapter of biomolecules of class 10th chemistry. Now, bio means living and molecules are substances which are made up of multiple atoms. Now, complex organic compounds which govern our day-to-day activities are known as biomolecules such as carbohydrates, proteins, nucleic acid, lipids, etc. Vitamins are also included here, along with certain minerals which are essential for the functioning of our body. Biology students have learned it in detail in class 11th. We will continue in class 12th on the chemistry aspect of it. First of all, carbohydrates, which is the most major part for your examination point of view. Carbohydrates are majorly produced by plants and are a form of very large group of naturally occurring compounds. Some are cane sugar, glucose, starch, etc. These are common examples. Now, most of the carbohydrates which you see around you have a general formula of CxH2yOy. For example, C6H12O6, and they are considered as hydrate form of carbon where the name carbohydrate was derived from. For example, the molecular formula of glucose C6H12O6 fits into this general formula C6H2OY. But all the compounds which fit into this formula may not be classified as carbohydrate. For example, rhamnose C6H12O5 is a carbohydrate but does not fit in this definition. Chemically, the definition of carbohydrates are optically active polyhydroxyaldehydes or ketones, or the compounds which produce such units on hydrolysis. Now, breaking down word and word, optically active are compounds which give you a mirror image based on it. Polyhydroxy means it contains multiple hydroxy OH groups and it will contain either one of aldehyde or ketones on the terminal ends or in the middle of the chain. Some of the carbohydrates are sweet in taste, that's why they are also now called as sugars. The most common sugar which is found in our home is sucrose, whereas the sugar present in milk is known as lactose. Now, classification of carbohydrates. Carbohydrates are classified on the basis of their behavior of hydrolysis, that is, how many units they yield on hydrolysis. Monosaccharides are the first type of carbohydrates. They cannot be hydrolyzed further and they are the simplest form of carbohydrates, for example, glucose, fructose, and ribose. Monosaccharides are further classified on the basis of number of carbon atoms and the functional group which is present within them. If a monosaccharide contains an aldehyde group, it is known as aldose, and if it contains a keto group, it is known as ketose. Number of carbon atoms constituting the monosaccharide is also introduced in the name, as in the example of evidences given. As you can see in the given table, based on the number of carbon atoms, different types of aldose and ketose are formed. For example, for a 3 carbon chain, it is known as triose, and it is commonly known as aldotriose or ketotriose based on the functional group present, aldehyde or ketone. Similarly, 4 for tetrose, P for pentose, 6 for hexose, and 7 for heptose. Now, oligosaccharides. These are carbohydrates which yield 2 to 10 monosaccharides units on hydrolysis. They are known as oligosaccharides. They can be classified as disaccharides, trisaccharides, tetrasaccharides, and moving on up to the 10 number. Depending on the number of monosaccharides they do on hydrolysis. Among these, the most common are disaccharides. The two monosaccharides unit obtained on hydrolysis of a disaccharide may be same or different. For example, on the hydrolysis of sucrose, one molecule of glucose and one molecule of fructose is obtained, whereas maltose only gives you two molecules of glucose which are identical in nature. Now, polysaccharides are carbohydrates which yield a large number of monosaccharides. Units on hydrolysis are called as polysaccharides. They can range from 10 to 11 to even thousands. Some common examples are starch, which is stored form of food, cellulose, which is found in plants, glycogen, which is found in liver, and gums, which are a byproduct from plants which we obtain. They are polysaccharides and they are not sweet in taste and are also known as non-sugars. Now, reducing and non-reducing sugars. The carbohydrate may also be classified as reducing or non-reducing. It is based if they get reduced by Fehling solution or Tollens reagent, which we have learnt about previously in the chapter aldehyde, ketone and carboxylic acid. All monosaccharides, whether aldose or ketose, are reducing sugars. That is, monosaccharides constitute of glucose, fructose and maltose. All of them come under the category of reducing sugars only. In disaccharides, if the reducing sugar or group is aldehyde or ketone group is bonded, they are non-reducing sugars. For example, sucrose. On the other hand, sugars in which functional group are free are known as reducing sugars. For example, maltose and lactose. Now glucose, we'll study in detail about it, which is also known as aldohexose. Aldo because it contains an aldehyde group and hexose because it is a 6-carbon molecule. Now there are two common ways of preparing glucose. Either we can do it from sucrose, which is also known as cane sugar. If we boil it in dilute acids in an alcoholic solution, it will give you glucose and fructose in equal amounts as a byproduct, as you can see here. And we can also obtain it from starch also commercially. Glucose is hydrolyzed by hydrolysis of starch and boiling with acidic nature. Here N can be any number ranging from 11 to 1000. Starch and cellulose are extremely large polymers made up of thousands of units, and those many molecules of water are needed to hydrolyze it into N molecules of glucose. The difference between glucose and fructose is simply that glucose contains an aldehyde group and fructose contains an ketone group as its primary functional group. Now, some common glucose reactions are given here. First is the oxidation of glucose. It can be done using Tollens reagent, where the aldehyde is oxidized into carboxylic acid. It can be done by Fehling solution, which gives you the same product. These are both known as gluconic acids. In the presence of bromine water, it also gets oxidized to gluconic acid. And in the presence of concentrated nitric acid, both the aldehyde and the alcoholic group get oxidized, which forms you saccharic acid. Next is the reduction reactions. First is the reaction with HI. In the presence of heating, glucose gets converted into N-hexane, which is also a test which is used to determine that glucose is a 6-carbon molecule. And on its reaction with sodium amalgam and water, the aldehyde group gets reduced to alcohol. Now, a note here, reduction with HI gives N-hexane, which shows that all the 6 carbon of glucose are arranged in a straight chain form. Now, oxygen formation. We have discussed about it in the chapter of aldehyde, ketone and carboxylic acid also. NH2OH, when reaction with the polar carbonyl bond, carbon being delta positive and oxygen being delta negative, oxime functional group gets attached to it. CHNOH is formed, which is known as glucose oxime. Next is cyanohydrins. Similar reaction is given with HCN also, which yields to the formation of chiral carbon, which is optically active. Both of these reactions on the aldehyde group have been covered in the chapter of aldehyde, ketone and carboxylic acid. Now, since this carbon has become chiral, so two different products will be formed, which are known as diastereomers. Now, acetylation is the reaction of adding acetyl chloride CH3COCl. All of the OH bonds present here would be replaced by CH3CO in this entire compound, which are 5 species, 4 in the middle and 1 in the terminal end. Next is the reaction with phenylhydrazine. It gets substituted in the similar manner as we have shown above. This is not part of your board examination syllabus, it is part of the competitive side. Now configuration of monosaccharides. Now glucose is correctly named as D plus glucose. D before it represents the configuration, whereas the positive sign represents whether it is dextrorotatory or levorotatory. Remember that D and L have no relation to optical activity of any compound. The meaning of D and L notation is given as follows. The letter D and L before the name of the compound indicate the relative configuration of a particular stereoisomer. This refers to the relation with a particular isomer of glyceraldehyde. Glyceraldehyde contains one asymmetric carbon atom and exists in two in an isomeric form. As you can see here, glyceraldehyde has aldehyde, hydrogen, alcohol and CH2OH group attached to it. If the OH group is on the right hand side, it is positive, and if the OH group is on the left hand side, it is taken as negative. The positive isomers which behave and have configuration similar to positive glyceraldehyde are known as D configuration, whereas those which correlate to the left hand side position of the OH group and the negative isomerism of glyceraldehyde are given the L type of configuration. These are important because you would need to give the proper type of configuration while giving the name of the compounds. For assigning configuration of monosaccharide, it is the lowest asymmetric carbon atom, which is shown above. As in positive glucose, OH on the lowest asymmetric carbon is on the right side, which is comparable to the positive of the glyceraldehyde. So this is assumed D configuration. Here you can see the configuration of glyceraldehyde and glucose side by side in the extended branch chain form. First OH molecule is present on the right hand side, then on the left, then the next two on the right hand side only. Right, left, right, right. This is the presence of OH groups in the glucose molecule in the extended form. Now this is the straight chain structure of glucose. Glucose can also exist in a cyclic structure, which we will discuss now. Glucose exists in two crystalline forms, which are named as alpha and beta. The alpha form of glucose is obtained by crystallization from concentrated solution of glucose, while the beta form is obtained by crystallization from hot and saturated aqueous solution. Both alpha D and beta D glucose undergo mutarotation in aqueous solution. Although crystalline forms of alpha and beta D glucose are quite stable in aqueous solution, but each form slowly changes into an equilibrium mixture of both. As you can see here, at a certain given temperature of 52.5 degree, we get an equilibrium position between both of the different isomers of glucose. Alpha D glucose and beta D glucose occur at different temperatures, and at their equilibrium point, it contains an equal mixture of both of them. This spontaneous change in specific rotation of an optical compound at equilibrium value is the term as mutarotation. Definition is important for your examination, to kindly remember it. When two different isomeric forms get rotated and attain an equilibrium mixture at a given temperature, then it is known as mutarotation. It was found that glucose can form a 6-membered ring in which OH at the 5th carbon is involved in the ring formation. This also explains the absence of CHO group and also existence of glucose in two forms, as you can show here. The OH from the 5th carbon is used here to form a linking between the 5th and the 1st carbon, and this forming of linkage forms such a structure with oxide linking here. Now, the two cyclic hemiacetal forms of glucose differ only in configuration of hydroxyl group at C1 called anomeric carbon, the aldehyde carbon before cyclization. Such isomers alpha and beta forms are called as anomers. The 6-member cyclic structure of glucose is called pyranose structure, because it resembles a lot from the molecule pyran in analogy to it. Pyran is a cyclic organic molecule, as you can see here. These are the cyclic structure of alpha D glucose and beta D glucose, as you can see here. The only major difference is the presence of hydroxyl group on the first carbon. Kindly remember these structures, they can be asked to be drawn in the examination. These are the steps if you want to draw the Haworth projection as above. Arrangement of atoms should be done in this manner. As you can see, oxygen links to the first and fifth carbon, and all others are in a hexagonal manner. And then you simply attach hydrogen in such a manner. First 2 hydrogen on top, then OH, then H, and then CH2OH in the upper part, and the lower part is simply similar to the straight chain as we have discussed before also. This completes your glucose part for this chapter, which constitutes the major portion of your board examination also, and the majority of your previous year questions also. We will continue with the other types of carbohydrates and different other biomolecules in the next parts. Thank you very much. Hope you enjoyed it. Continue learning and continue studying.